Welcome to this lesson on DNA bases. DNA, or deoxyribonucleic acid, is the molecule that carries genetic information in all living organisms. The building blocks of DNA are called nitrogenous bases, and understanding these bases is fundamental to understanding how genetic information is stored and transmitted from one generation to the next. Nitrogenous bases are nitrogen-containing molecules that form the core structure of nucleotides in DNA. Think of them as the letters in the genetic alphabet. Their primary function is to store and transmit genetic information through their specific pairing patterns. These bases are called nitrogenous because they contain nitrogen atoms in their molecular structure, which makes them chemically distinct and capable of forming specific bonds with each other. DNA contains exactly 4 different nitrogenous bases. These are adenine, represented by the letter A, thymine, represented by the letter T, guanine, represented by the letter G, and cytosine, represented by the letter C. These 4 bases are often abbreviated as A, T, G, and C, and every piece of genetic information in your DNA is written using just these 4 letters. The sequence in which these bases are arranged determines the genetic instructions for building and maintaining an organism. One of the most important principles in DNA structure is base pairing. Adenine always pairs with thymine. This is not a random occurrence, but a result of their complementary chemical structures. The shape and chemical properties of adenine perfectly match those of thymine, allowing them to bond together. This specific pairing is crucial for DNA replication and the accurate transmission of genetic information. Similarly, guanine always pairs with cytosine. Just like adenine and thymine, guanine and cytosine have complementary structures that allow them to fit together perfectly. These 2 base pairs, A with T and G with C, form the rungs of the DNA double helix ladder. This complementary base pairing is what allows DNA to replicate itself accurately and maintain genetic information over many cell divisions. Let us review the base pairing rules. Rule number 1, adenine always pairs with thymine, written as a -T. Rule number 2, guanine always pairs with cytosine, written as G-C. Rule number 3. These pairs are complementary and form the DNA double helix. This means that if you know the sequence of bases on one strand of DNA, you can determine the sequence on the other strand because of these complementary pairing rules. The base pairs are held together by hydrogen bonds, which are relatively weak chemical bonds. However, there is an important difference between the 2 types of base pairs. The -T pair is held together by 2 hydrogen bonds, while the G-C pair is held together by 3 hydrogen bonds. This means that G-C pairs are slightly stronger than -T pairs. This difference in bond strength has important implications for DNA stability and the energy required to separate the 2 strands. Let us summarize what we have learned. First, there are 4 bases in DNA: adenine, thymine, guanine, and cytosine. Second, these bases follow complementary pairing rules. A pairs with T and G pairs with C. Third, hydrogen bonds hold base pairs together, with a -T having 2 bonds and G-C having 3 bonds. Finally, this base pairing is fundamental to DNA structure and replication, ensuring that genetic information is accurately copied and passed on. Understanding these basic principles is essential for learning more advanced topics in genetics and molecular biology.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.